Sds-Page, sodium tiodecyl sulfate, polyacrylamide gel electrophoresis, is an electrophoresis method and very powerful technique in the study of proteins. In Sds-Page, proteins are separated in polyacrylamide gel based on their molecular weight. For sample preparation, a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein samples. Protein sample may be biologically derived, for example, from prokaryotic or eukaryotic cells, tissues, viruses, environmental samples, or purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amide and carboxyl functional groups along with a side chain. Proteins are formed by linking amino acid with peptide bonds and fold into one or more specific spatial conformation, driven by a number of interactions such as hydrogen bonds, hydrophobic interaction, disulfide bonds, and ionic bonds. SDS is an anionic surfactant and contains a polar head group with a negative charge at the end of a long hydrophobic carbon chain. SDS denature the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interaction, while the reducing agent, beta-mercaptoethanol, is used to cleave the disulfide bond. Also, when a protein mixture is heated, SDS bind uniformly to the proteins, and the intrinsic charges of these proteins become negligible when compared to the net negative charge contributed by the SDS. This treatment brings the folded proteins down to the linear molecules with net negative charge. Therefore, this protein can be separated in a polyacrylamide gel based on their molecular weight.
For gel preparation, a separating gel solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate, and timid. The gel is produced by polymerization between two glass plates tankered vertically in a cassette. The separating gel is poured first. The polymerization reaction is initiated by a free radical generating system. Ammonium persulfate is used as a free radical initiator, while the timid accelerates the rate of formation of free radicals from persulfate. These free radicals convert acrylamide monomers to free radicals, which react with an activated monomers to begin the polymerization chain reaction. The elongating polymer chain are randomly crossing bisacrylamide, resulting in a gel with a characteristic porosity. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight samples. The stacking gel solution is poured on top of the solid separating gel, and a plastic comb is placed on the top of the stacking gel during polymerization, enables the formation of small wells in the gel. Once the gel completely polymerizes, the comb is removed and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel.
The gel is inserted into a chamber and a trace glycine chloride buffer system with pH 8.3 is poured to allow the conduction of the current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the protein were denatured, they stayed at rest throughout the run. Pre-sample application. In addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of a protein of known sizes and thereby allow the estimation of the sizes of the protein in the actual samples, which migrate into parallel in different tracts of the gel. Each sample is perpetuated into its well in the gel, and we have seen previously bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is used for visualizing the sample in the well, and glycerol increases the density of the sample and used to make the sample fall to the bottom of the sample well, rather than just flow out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel.
causing the negatively charged proteins to migrate across the gel away from the negative electrodes and towards the positive electrodes. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel. The stacking gel is used to ensure the walls of the proteins arrives at the separating gel at the same time because the gel wells are around 1 cm deep. So in the absence of the stacking gel, the proteins would all enter the separating gel at the same time, resulting in a very smear balance. As we have seen previously, the running buffer consists of the glycine and chloride ions. So in the sample well, we will have the proteins, the chloride ions, and the glycine. Proteins and chloride ions are negatively charged, while glycine can exist in 3 different charge states depending on the pH. The isoelectric point pH of glycine is 5.97. At this pH, glycine carries neutral charge due to gain and loss of protons. At a pH below its pHi, glycine carries a net positive charge, and at pH above its pHi, it becomes negatively charged.
At this pH, glycine is negatively charged. Once the electric current is applied, proteins, chloride ions, and glycine are forced to enter to the stacking gel, where the pH is equal to 6.8. At this pH, glycine switches predominantly to neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride ions migrate in front of the protein as leaving ions. Glycine molecules migrate behind the protein as initial trailing ions. Consequently, proteins are trapped to the concentrated neural zone between the chloride ion and the glycine. This procession carries on to locate the separated gel where the pH reaches to 8.8. At this pH, glycine molecules are mostly negatively charged and can migrate much faster to the proteins. Proteins are separated as molecules with higher molecular weight. Protein moves so slowly through the pores of polyacrylamide gel electrophoresis. The lower molecular weight proteins, due to the small molecules size of pronational blue, migrate much faster than proteins, and by optical control of the migrating color band, the electrophoresis can be stopped before they die and before the sample has completely migrated through the gel. At the end of the electrophoretic separated, all proteins are sorted by size and can then be analyzed by other methods. For example, protein staining method and neurological detection, such as the Western blot technique. After removing the class split, stacking gel is regarded as the separated gel and can be stained. Compassive blue is the most popular protein stain. It is an anionic dye which is non-specifically bound to proteins. The structure of the compassive blue is predominantly nonpolar, and it is usually used in methanol solution with acidified acetic acids. Proteins in the glycine fixed by acetic acid are simultaneously stained. After the staining, the proteins are detected as a blue band on a clear background. The molecular weight of size marker in a separated plane in the gel can be used to determine the approximate molecular mass of the unknown biomolecule by comparing the distance traveled relative to the marker.
Agarose gel electrophoresis is a technique used to separate DNA fragments or other macromolecules such as RNA and proteins based on their size and charge. The first step for DNA fragment separation is the gel preparation or gel casting. An appropriate amount of agarose powder is weighed out. For standard agarose gel electrophoresis, larger molecules are resolved better using a low concentration gel, while smaller molecules separate better at high concentration gel. Once the agarose has been weighed, an appropriate buffer such as the TAE or TBE is poured into the bottle. Agarose does not dissolve until it is heated. Therefore, the solution is heated for a few seconds in a microwave oven. Once the agarose is fully dissolved, the solution is cooled to about 55 degrees Celsius in a water bath.
While the agarose solution is cooling, an electrophoresis tank with a negative electrode and a positive electrode is prepared for gel casting. The comb is placed in the tank to create wells for loading samples. Then casting dams are fixed in the tank so the Agarose solution does not flow out during setting. Once the Agarose solution must have cooled to about 55 degrees Celsius and before pouring it into the electrophoresis tank, ethidium bromide is added. The ethidium bromide is highly toxic, so it must be carefully handled with a firm hold.
Ethidium bromide is an interlacing agent which interlates between base pairs of the DNA molecules when it is exposed to ultraviolet light. It will fluoresce and the DNA becomes visible. The more DNA present, the brighter the band. The agar solution is then poured into the fluorescent tank. Then the gel is allowed to solidify at room temperature. Once the gel is solidified, the comb is carefully removed as well as the casting bands. Next, the gel is submerged in TAA, TAE, and TBE, running buffer, which is used to provide ions to maintain the pH of the relatively constant value.
The next step is the preparation of the DNA samples. A loading buffer is added to each sample. This buffer contains glycerol and certain dyes, such as bromophenol blue. It is used to add density to the sample, allowing it to sink into the gel. It also provides color to simplify the loading process and helps the user monitor the progress of the separation. Before loading the samples into the wells, a molecular weight site marker, known as a DNA ladder, is commonly used. The DNA ladder is a standard reference that contains DNA fragments of known length. It is loaded into the first well of the Agarose gel. Once the molecular weight marker is loaded, the DNA samples are then loaded into the wells. After the samples are added, a lid is placed on the electrophoresis tank. Then an electric current is applied to pull the samples through the gel based on their charge and size.
The DNA molecules will travel through the gel with different speeds. The DNA molecule have a negative charge because of the phosphate group and the sugar phosphate backbone. Therefore, when placed in an electric field, the DNA fragments start moving through the matrix of the gel towards the positive pole because all DNA fragments have the same amount of charge per mass. Small fragments move through the gel faster than large ones due to the relatively small molecule size of bromophenol blue. It migrates faster than DNA, and by optical control of the migrating colored band, the electrophoresis can be stopped before they die. So before the samples have completely migrated through the gel and left. Once the DNA fragments have been separated, the gel is placed on the UV light. Then the DNA fragments can be seen as bands. Each band contains a large number of DNA fragments of the same size that have all traveled as a group to the same position. By comparing the DNA bands of the sample to the DNA ladder, we can determine their approximate sizes.